AIDS is a real, bona fide disease. Its etiology is a virus, which we call HIV. Its pathogenesis is the process by which the cells that are infective have certain changes uh, and certain periods of latency, but ultimately result in progressive T cell loss, as well as many, many other cells besides lymphocytes being infected. It has a wide variety of morphologies, most of which are the result of secondary opportunistic infections, and clinical expressions of the disease are the same as the morphology. It's a wide variety of opportunistic infections, neoplasms, progressive immune failure, death often, uh, and expression of the viral RNA in the blood called the viral load. Epidemiology of AIDS has changed a bit. In general, the heterosexual causes are on the rise. The homosexual causes are on the decline. The intravenous causes, still big and number two, have remained about the same, haven't changed very much. The virus itself is the etiology of the disease, and there are a wide variety of antigens coded by generally genes, which we'll talk about. The GP120 antigen, little tip of the ball here, is always the most famous one. The stalk connecting the GP120 to the virus is GP41. The matrix is underneath that, often termed P17. There's a capsid underneath that, often called B24. There's protease, which, as you might guess, would be involved in the cell's invasiveness or the ability to chew through human cells to do what it has to do. There is the enzyme called reverse transcriptase as part of its genome, which does the opposite. That's why it's called reverse transcriptase, because it's the process from which RNA goes to DNA rather than vice versa, which you normally see in transcription. In the pathogenesis of the disease, we have viruses which attach to cell membranes. They could be T cells, they could be lymphocytes, but they could really be any cell, and eventually result in the virus doing its thing in the cytoplasm, budding off new virion particles, and then these, of course, traveling on to infect more cells. Here is a picture of an early bud, which looks like no more than a membrane thickening on a transmission electron microscopy here. Here's another real nice picture of late budding, and here's some spectacular pictures of the free virions, perhaps freshly made. The basis for most of the therapy of AIDS in the early days were drugs that would be able to inhibit reverse transcriptase. Let's define reverse transcriptase properly. It's an enzyme. It's used by the retrovirus to transcribe single-stranded RNA into single-stranded DNA and subsequently construct a complementary strand of DNA, providing a way from which a RNA is now transcribed to DNA. So that's why it's called reverse transcriptase. If you look at the various common genes, part of the genome in the HIV virus, and of course HIV virus is a redundant term, isn't it? There is a GAG gene, there is a POL gene, POL, there's VIF, VPR, and these are all the primary parts of the genome which encode for the various viral protein areas we talked about, including polymerases, including proteases, including reverse transcriptase. So if you hear GAG, POL, VIF, VIPR, VUPU, and these are all genes which are coding for portions of the HIV protein. Clinically, pathogenesis of the disease results in a primary infection through blood, sex, saliva, uh, followed by a lymphoid infection and an acute uh, viral type syndrome, uh, so-called prodrome effect, within several weeks after the primary infection. There is an immune response. There's a long latency period, often many years. 7, 10 years is about average. And then by the time the expression of full-blown AIDS, so to speak, happens, we then see a variety of opportunistic infections based on total failure of the immune system. And I would be really happy if you thought of these processes linearly, because you can't get a lymphoid infection unless you've had a primary infection, and you can't get an acute syndrome unless you've had a lymphoid infection, and so on and so forth. And what we call AIDS, of course, is at the very end of the pathogenesis. Here's a nice diagram in which we have the primary infection, the lymphoid infection, the acute syndrome, the latency, and full-blown AIDS. If you would like to look at that in terms of graphs, if you would like to look at the dotted blue line here as representing the clinical latency, you can see that from the, let's say, 6 weeks from the acute syndrome, from the beginning of the infection, we have a period which you can see lasts an average of about 7 or 8 years before the rise of clinical symptoms, resulting in opportunistic infections and then often in death. So if you looked at some of the antibodies involved with AIDS, the anti-P24 antibody probably starts to appear, let's say, an average of 8 to 9 to 12 weeks after infection, and it stays up for many years. There's an anti-envelope antibody that appears even quicker and stays up even longer. And then there are just various viral particles, which is represented by this red line, which of course has a rapid peak during the viral prodrome and then stays very, very low levels until, of course, full-blown AIDS happens, and then they rise rapidly. This is when you're apt to get your greatest increases in RNA, or viral RNA. Some of the general immune abnormalities include lymphopenia. Why? Because the lymphocytes are being destroyed. Decreased T cell function. B-cell clonal polyclonal activation, resulting in increased expression of a variety of antibodies. And of course, not only are the T cells decreased in function, but so are your monocyte macrophage functions as well. The common opportunistic infections with AIDS: Cryptosporidium, PCP, toxoplasmosis, Candida, TB, nocardia, and a whole wide variety of herpes virus type viruses, some of which are very, very, very rare unless you are in severely immunocompromised patients. PCP was very, very rare until the AIDS epidemic came around, and now you can assume that every AIDS patient carries it. Toxo was considered to be rare as well. Very, very common effect, especially CNS in AIDS patients. PCP could express itself as the radiologist will call it a cotton wool or woolly infiltrates throughout the lung fields. And coincidentally, the pathologist will also use the word wool when referring to microscopically woolly or cottony type substances in the alveoli, which, if they are stained with silver stain, you can see the actual pneumocystis organisms. Cryptosporidium, an organism which pretty much adheres to the epithelial cells of the large bowel, can cause dysfunction of that, and of course the main symptom would be diarrhea. TB, or caseating granulomas, as you see here, very, very, very common with HIV patients. Here's some multinucleated giant cells. Here's some caseation or necrosis in the middle of the granuloma. And here you could see a little lymphocytic cuff of reaction around the histiocytic clusters or macrophage clusters, which is the granuloma. Kaposi sarcoma, B-cell lymphomas, CNS lymphomas, cervix cancer, squamous cell, all the result of AIDS. And in most cases, it's really the result of other viruses, like HPV going rampant when it can't be fought off properly. CNS lymphomas were basically unheard of until AIDS came around, and now it's probably the first or second most common cause of a space-occupying brain lesion in an AIDS patient. A wide variety of B-cell lymphomas, and of course Kaposi sarcoma, not caused directly by the AIDS virus, but a herpes virus, which is allowed to run rampant when there's no immune protection. I think we'll end the discussion here and finish up with amyloidosis as the last segment of this chapter. Thank you very much.